Embryology of the male external and internal genitalia. The development of the male external and internal genitalia is a highly complex process that begins early in embryonic development. It is controlled by both genetic and hormonal signals, primarily the presence of the Y chromosome and testosterone. Here's a detailed overview of how the male genitalia develop embryologically. First, chromosomal determination of sex. The development of male or female genitalia is determined by the genetic sex of the embryo, which is established at fertilization. XY chromosomes. Male. If the sperm carries a Y chromosome from the father and fertilizes the egg, the resulting zygote will be male, XY. The SRY gene, sex-determining region Y, on the Y chromosome plays a crucial role in initiating the development of male sexual characteristics. XX chromosomes. Female. In the absence of a Y chromosome, XX combination, the embryo develops female sexual characteristics. 2. Early gonadal development, weeks 4 to 7. Indifferent gonads. In the early stages of embryonic development, around week 4, the gonads, testes or ovaries, are undifferentiated and called indifferent gonads. These gonads have the potential to develop into either testes or ovaries, depending on the presence or absence of the SRY gene. Testicular development. If the SRY gene is present, it triggers the formation of the testes from the indifferent gonads. The testes begin to develop around week 6, and by week 7, they produce testosterone, a key hormone that influences the development of male genitalia. Ovarian development. In the absence of the Y chromosome, the gonads develop into ovaries, and female reproductive structures begin to form. Third, formation of male internal genitalia. Around the 7th week of development, the male internal genitalia begin to form under the influence of testosterone produced by the developing testes. Male internal genitalia, key structures, Wolffian ducts. These are the precursor structures that will develop into the male internal reproductive organs. In the presence of testosterone, the Wolffian ducts develop into the epididymis, vas deferens, and seminal vesicles. The Wolffian ducts are initially paired structures that form near the kidneys. Testosterone promotes the development of the vas deferens, epididymis, and seminal vesicles, which are crucial for sperm transport and storage. Mullerian duct regression. In females, the Mullerian ducts develop into the uterus, fallopian tubes, and upper portion of the vagina. In males, the Mullerian ducts regress under the influence of anti-Mullerian hormone, AMH, produced by the Sertoli cells in the testes. This prevents the development of female reproductive organs. Formation of prostate and bulbourethral glands. The prostate and bulbourethral glands develop from the urethral and paraurethral tissue influenced by androgens, like testosterone, that direct the formation of these structures. The prostate develops from the urethral buds, and the bulbourethral glands arise from urethral tissue near the base of the penis. Fourth, formation of male external genitalia, weeks 8 to 12. The external genitalia begin to form between the 8th and 12th weeks of pregnancy, and this process is also regulated by testosterone and dihydrotestosterone, DHT, a more potent form of testosterone. Male external genitalia development. Genital tubercle. Early in embryonic development, both male and female embryos have a genital tubercle, a small protuberance between the legs. The presence of testosterone stimulates the genital tubercle to elongate and develop into the penis. Urogenital sinus and fold. The urogenital sinus and the genital folds form from the primitive urogenital structure and play a key role in the development of the male external genitalia. The genital folds fuse to form the urethra within the penis, a process that is completed by the 12th week of development. In males, these folds also form the scrotal swellings, which will develop into the scrotum. Scrotal development. The scrotal swellings on either side of the genital tubercle fuse to form the scrotum, which will house the testes. The process is influenced by androgens, particularly testosterone. The descent of the testes into the scrotum occurs later in fetal development, usually between the 7th and 9th months of pregnancy, though some males may experience a delay in this descent, a condition known as cryptorchidism. Penis formation. The genital tubercle elongates and differentiates into the shaft of the penis, while the urethral groove along the underside of the tubercle closes, forming the urethra. Dihydrotestosterone, DHT, produced from testosterone, is a key hormone responsible for the complete masculinization of the genitalia, including the development of the penis and scrotum. Fusion of the urethral folds. The urethral folds, which initially were separate, fuse along the ventral aspect of the genital tubercle to form the penile urethra. The external opening of the urethra forms at the tip of the glans penis by the end of the 12th week. 5. Descent of the testes. By the 7th to 9th month of fetal development, the testes, which initially develop in the abdomen near the kidneys, descend into the scrotum through the inguinal canal. This process is controlled by androgens and involves the action of the gubernaculum, a band of tissue that helps guide the testes into the scrotum. Testicular descent. The testes follow a path through the inguinal canal into the scrotum, where they will be housed for sperm production. The descent of the testes is critical for normal sperm development, as the scrotum maintains a temperature lower than the rest of the body, which is necessary for spermatogenesis. 6. Hormonal influence. The development of the male internal and external genitalia is regulated by several hormones. Testosterone, produced by the Leydig cells of the testes. Testosterone is the key hormone that drives the development of the male reproductive structures, including the Wolffian ducts, penis, and scrotum. Dihydrotestosterone, DHT. This more potent form of testosterone is essential for the masculinization of the external genitalia, including the formation of the penis and scrotum. Anti-Mullerian hormone, AMH. Produced by Sertoli cells in the testes, AMH induces the regression of the Mullerian ducts, preventing the formation of female reproductive organs. Inhibin, secreted by Sertoli cells. Inhibin helps regulate the function of the hypothalamic pituitary gonadal axis, particularly in the regulation of sperm production. In conclusion, the development of male internal and external genitalia is a complex process that is primarily governed by genetic factors and hormonal signaling. Testosterone and dihydrotestosterone play crucial roles in the differentiation of the Wolffian ducts into male reproductive structures, the formation of the penis and scrotum, and the descent of the testes into the scrotum. This process begins early in embryonic life and continues through fetal development, ensuring the proper formation of male reproductive organs.